Okay, well, welcome everyone to Tumor Dormancy with Dr. Lyerly. My name is Christine Benjamin, and I'm the Breast Cancer Program Director here at SHARE. Before the presentation begins, I'd like to tell you just a little bit about SHARE. SHARE is a 40-year-old nonprofit organization that helps people through breast or ovarian cancer, offering the support of those who've been there. SHARE offers many services, including helplines, telephone and in-person support groups, and educational programs. All services are free of charge to participants. For more information, you can visit our website at sharecancersupport.org. Now, Ivis Sampaio, our Senior Director of Programs, will introduce Dr. Lyerly.
Hello, everyone, and welcome. Dr. Lyerly is the George Barth Geller Professor of Cancer Research, Professor of Surgery, Associate Professor of Pathology, and Assistant Professor of Immunology at Duke University in North Carolina. He was appointed in 2008 by President George Bush to serve a 6-year term on the National Cancer Advisory Board, which oversees the U.S. National Cancer Institute. He served as the chair of the Cancer Centers Subcommittee and on the Global Health Subcommittee of the National Cancer Advisory Board. He has also served on the National Institute of Health Council of Councils and the board of the National Institute of Health Office of AIDS Research. A surgical oncologist, Dr. Lyerly served as the PI of the Duke Breast Spore Program and as the director of the Duke Comprehensive Cancer Center from 2003 to 2011. Dr. Lyerly is an internationally recognized expert in cancer therapy and immunotherapy and has published over 300 scientific articles and book chapters and has edited 10 textbooks. He serves on the editorial board of 12 scientific journals. Dr. Lyerly is involved in cancer research and education internationally and has research collaborations in cancer care, cancer immunotherapy, and cancer genomics with faculty members in China, India, and Pakistan. He has been a faculty member of the Medical Oncology Group of Australia Clinical Trials Workshop and has conducted clinical trial workshops in Singapore, China, India, Nepal, and Pakistan. I'm honored to have Dr. Lyerly presenting today on our webinar, Tumor Dormancy.
Well, thank you very much. This is Kim Lyerly, and I'm delighted to be here as a part of this SHARE webinar on tumor dormancy. I know that this is an important topic for many people, and I'm happy to address any questions or thoughts during the course of the webinar, and again, invite those who are participating to provide their thoughts. Let me get started by describing one of the challenges we face as clinicians, and what I'm going to show you is a slide that represents the survival rates of breast cancer patients over a 20-year period of time. And this was work published a few years ago. But what is interesting about it is that it describes the molecular subtypes of breast cancer that are recognized today and the overall survival of patients with these molecular subtypes. So I draw your attention to the green diamonds. And you can see at 5 years there's a significant drop-off in survival in patients who have the green diamonds, which actually represents triple-negative breast cancer, or ER, PR, HER2-negative breast cancer. Many people feel that this is the most aggressive and most lethal form of breast cancer, but as you can see, there is a big drop-off. But at 10 years, there is a persistent drop-off of survivors. But at 20 years, it appears to plateau or stabilize. So the number of survivors at about 20 years is very similar to the number of survivors at 10 years. In contrast, if you look at the light blue diamonds, at the 5-year interval, we see that there's an overall survival that was superior to the triple-negative breast cancer. But at 10 years, there is an incremental decrease in survivors. Again, very similar to the triple-negative breast cancers in that decrease. But over the period between 10 and 20 years, you see a persistent drop in survivors with ER and PR positive breast cancers. So at the 20-year interval, strikingly enough, the number of survivors with the ER-PR positive breast cancers is less than with triple-negative breast cancers. So the conundrum here is, why is it that after treatment at 5 and 10 years, we have better survivors, better survival rates with the ER-PR positive cancers, but if we look over the 10 to 20 year period of time, we see an ongoing decrease in survival. And many people have suggested, perhaps, and I've included an animation, that this difference between the survivors at 5 years after they finish their treatment and the survivors at 20 years is perhaps the reactivation of what are considered dormant tumor cells. These aren't new breast cancers that are forming, but perhaps tumor cells that have found a way to reside in your body and become reactivated, ultimately causing your death. Now, as we point out, in triple-negative breast cancer, it is a significant risk of death occurring in the first 5 years. But in contrast, there does not seem to be a dormant state for the triple-negative breast cancers because they tend to be plateauing in that their survival at 10 years is very similar to their survival at 20 years. So this has led many people to suggest that perhaps there is a phenomenon of dormancy. Now, again, in a graphic form, this biology is depicted here in this graph from one of the Nature reviews. The tumor burden is the graph on the x-axis, and then the y-axis is over time. You can see from your left, there's a growth of the tumor cells until it becomes clinically evident. Oftentimes, people will have surgery or some kind of excision of the primary tumor that reduces the tumor burden, and adjuvant therapy is designed to eliminate and kill micrometastatic disease or these potentially disseminated tumor cells. Unfortunately, as we see in the dotted black lines, that there are some individuals who will have a recurrence after some period of time. And again, one of the questions we have is that a dormant tumor cell? Was that tumor cell somehow resistant to the adjuvant therapy, whether that's chemotherapy or hormonal therapy? So scientists have been trying to study this phenomena, and I think it's pleasing to be able to report significant advances in the science behind what's happening. And this cartoon illustrates some of what is now known about the biology of dissemination and metastases leading to what we call colonization. So on your left, there are cells of the primary breast cancer that will enter into the circulation. And there's now an understanding of the mechanism in which those cells can leave the circulation. In the cartoon at A, you can see a mechanical trapping. If the blood vessels become increasingly smaller as they form capillaries and so forth, the tumor cells can then get stuck to the side of the vessel. B is there might be a biochemical signal for the tumor cells to stop. And then C, it could be that there are areas just outside of the blood vessels that create a niche or a home for these cells to reside. And these three processes that lead to the tumor cells to leave the blood vessels create an opportunity for tumor cells to become quiescent. And again, that definition is not as clear as we would like, but there is a set of evidence that provides some support that the cells are quiescent at a time, and then once they've passed through that quiescent stage, if you look at the cartoon and see a micrometastasis could form, meaning that the tumor cells are beginning to proliferate and form a very small nodule of tumor that's not detectable clinically by X-ray or by blood test. And then eventually these micrometastases will form larger tumors, and these larger tumors are the form of metastatic breast cancer that require treatment, and they have the potential to cause great harm, even death. They can cause bone fractures, they can replace your liver, so you eventually have liver failure. They can get into your lungs and cause you to have pneumonia and die, et cetera, et cetera. So significant morbidity and death from the macrometastatic process. So this has led, again, scientists to try to understand what are the opportunities to intervene, and again, do we know enough to really do something about this process? For example, one question is, how do the breast cancer cells get to the bone specifically? What is it about the circulating tumor cells that allows them perhaps to preferentially go to bone? And if they're in the bone, where do they actually exist? Is there an X-ray or a blood test or some examination that can tell you that you have these small micrometastatic lesions in your bone? And if they are in the bone, and there's some evidence that they are in the bone at the time of your diagnosis, very, very early in the process of having breast cancer, is there some intervention that can be developed that will prevent them from ever developing into micrometastases? So in this next cartoon, I'm going to tell you and give you some insight into what we know about this process, and actually it reflects the biology of our understanding how any cells leave the bloodstream. And much of this work was done in trying to understand how inflammatory cells or cells of the immune system leave blood vessels to become effector cells to fight infections or cause inflammation and so forth. And the title should be, How Do Malignant Cells Get into the Bone or the Bone Marrow? Sorry, there's a typo there. And this cartoon shows that blood white cells, they don't just get through the tissue by randomly bumping into blood vessels, but there are very specific receptors and receptor ligands that are expressed by the lining of the blood vessels, and these lining receptor ligand pairs are called E-Selectins, and these E-Selectins act as brakes to allow blood cells that are in the circulation that are going by like in a river. These brakes attach to the blood vessel wall, the white cell, and then the white cell can slow down enough to make its way through the blood vessel wall and enter into the stroma or extravascular space. Now what investigators have found is that breast cancer cells also express these receptors that can act as brakes, and the breast cancer cells that have large amounts of these brakes on them in fact do correspond with tumors that have a much higher likelihood of recurring late. So the idea is that these receptors slow down white cells so they can extravasate or leave the blood vessel, and these same receptors are found on metastatic or late recurring breast cancer cells. Now, as we pointed out, the cells that leave the blood vessels they have a number of pathways that can lead to their removal. They can simply stop growing and die, but they also may exist by having controlled growth followed by balanced apoptosis, meaning there's some death of cells as well as some growth. Some people talk about leaves growing and falling off on a cyclical basis. And if you can imagine a population of tumor cells that will grow slowly and some of the cells will die, you might have a situation in which a very small amount of tumor cells may appear quiescent because they are just slowly cycling. Another proposed set of activities is that the tumor cells may grow, but they may not grow new blood vessels, and without blood vessels and the nutrients from blood vessels, you control the magnitude of growth that you can achieve. So basically, if you imagine something growing and needing nutrition and not having a source of nutrition, you end up having a nutrition-limited, if you will, mass of tumor cells. And then on your left side, the idea perhaps is that maybe the immune system is actively controlling these micrometastases, meaning that there is an immune response to your tumor, and part of your immune system is killing the tumor cells that are trying to grow while other tumor cells are growing. Again, there is some evidence that this is happening, but the quality of the immune response and the magnitude of that immune response versus the ability of the tumor cells to escape immune recognition or to be stealthy and avoid recognition is something that people are quite interested in exploring. So when we think about dissemination, as I point out the different scenarios that people are envisioning, it's somewhat complicated by the fact that, again, in this cartoon on the left, we say that the tumor cells leave the blood vessel, and then the question is, do the cells themselves stay in a quiescent stage, meaning that they have intrinsic cellular properties? They're vampires, if you will. They're frozen in time, and they're going to reactivate at some point in the patient's life. Or, as we described earlier, is it a population effect, meaning any one tumor cell is either growing slowly or going through apoptosis or being eliminated by the immune system. So the end result is that there's a small population that's dynamically changing but never growing in size. And then eventually you develop either a breakthrough that leads to macrometastasis and ultimately recurrence or death of the patient, or that you have an ongoing state of dormancy either at the cellular level or the population level. Now this is very important because if it's an intrinsic feature of the tumor cell, then the only way to manage that is to develop a medicine or a treatment that will directly affect the tumor cell. But if it's a population effect, you can imagine anything that would disrupt the dynamics of that population could in fact be a way to treat micrometastases or disseminated or dormant tumor cells. So the questions are quite important to answer. Now, how do we get there, and are there examples in nature in which dormancy exists? And it turns out, oddly enough, that our understanding of stem cells and the biology of stem cells and their natural history appears to be quite similar to this, this sense of dormancy. And the reason is that dormant stem cells reside in your body. They reside, in fact, for your whole life. And these stem cells have the potential to repopulate many organs and compartments of your blood system, of your skin, Many people think that stem cells are part of almost every organ, and there is the capacity to regenerate significant numbers of cells from almost any organ. For example, livers can be significantly damaged or even surgically resected, and there's a significant regeneration of your liver. Many people feel that this is, from a hepatic or liver stem cell. So if we could understand the natural processes in which stem cells lay quiescent and become reactivated to become normal cells, is it likely that tumor cells also use many of these same molecular tools to become dormant or to reactivate? And on this cartoon, you can see on the upper side of the panel the stem cell regulation, the quiescence, the activation signals, proliferation, and differentiation are being studied by many, many groups around the world. And on the bottom half of the panel, we are recapitulating those events, but instead of having normal stem cells that are necessary for our health, we're looking at dormant, disseminated tumor cells that are laying sort of stealthily in our bodies, ready to be reactivated to proliferate and form metastatic disease that are going to be lethal for us. And are there tumor-specific signals that we can identify and use to intervene in preventing this from happening? So as we've talked before, by using the stem cell niche model and understanding what are the molecular pathways for stem cells, we are applying those insights into what causes a stem cell to be dormant and to be reactivated, and using those same tools to interrogate what's happening in disseminated tumor cells. And this is just an example of the types of cells and the environment in which a cell can live that can be influenced. And these cells represent hematopoietic stem cells, examples of a dormant disseminated tumor cell, and other cells that reside in bone marrow and the perivascular tissue, including neurons and osteoblasts, etc. Again, these things are quite important because some people have recognized that periods of stress can lead to recurrences, and some people have felt that there's a relationship between the nervous system and the capacity to have a recurrence, and the molecular pathways in which these signaling pathways can converge on disseminated tumor cells through hormones or stress hormones or neurons. They can see these connections by looking at the variety of cells that reside in the bone marrow. Now, what I'm going to do now is give you a slight peek into some work done by Dorothy Simpkins, who I was a collaborator with on this paper that was recently published in a journal called Science Translational Medicine. And you can see from the cover story, the takeaway here is that cancer cells are shown to migrate from blood vessels into parts of the bone marrow, and there's some early data that suggests that there's enough known about this pathway that we can actually begin to target it and either prevent those cells from lying dormant, or if they are lying dormant, can we get them to release themselves and get back into the circulation and become more apparent. And let me just show you some figures from this paper that might provide some insight into the kind of studies that are being done. So as we mentioned earlier, E-Selectins are critical to allow white blood cells to leave the blood vessels, and we know that E-Selectins are expressed on breast cancer cells. Again, if E-Selectins are present everywhere, why don't blood vessel cells just cause white cells and tumor cells to stick to their surface. But in this example, you can see this, this animation. We have the blood vessels shown here in the red, but then I'm going to show you the E-Selectin expression, or where the E-Selectin molecules are. And they're not in the central blood vessels, but they're in these microcapillaries and these blood vessels that are almost like small parking lots on the side of the major blood vessels. So you can see that a blood vessel cell or white blood cell that does not have E-Selectin on it will just pass through the bloodstream, continue its circulation. And in fact, a tumor cell, if it had high levels of E-Selectin, would just pass on through. But if the E-Selectin level and the ligands and receptors for E-Selectin were present on that breast cancer tumor cell, it would find its way into these parking lots, and these parking lots we found are present in the bone marrow. Again, this is a synthetic view where we have the entire blood vessel and E-Selectin populations, and you can see, rather than being a uniform set of signals, these parking lots appear to be opportunities for tumor cells to reside. This next slide shows you, in fact, evidence of this in an experimental animal model where we have a mouse that has circulating metastatic, if you will, breast cancers that are ER positive, and we inject them into the mice. They go through the circulation, but these little red cells are the metastatic breast cancer cells. And just as you saw before, the parking lots were not in the central core, the central vessel, but in the sinusoidal region. And in fact, you see that's where these metastatic breast cancer cells end up residing, and these MCF7 cells do have that E-Selectin receptor. And in further support of that, we can see if we take bone marrow samples from patients themselves, again, some of these receptors, we call one SDF1. On the top two panels of this slide, we see these tumor cells that are disseminated. They're in the bone marrow. They have little arrows pointing to them. And so they're present. They are in the little parking lots next to the blood vessels. And then on the bottom two examples, you see a marker of cell proliferation, Ki67. If these cells were proliferating and growing the way typical aggressive breast cancers were, metastatic lesions, etc., they would be Ki67 positive. But in fact, these tumor cells are expressing these E-Selectin type ligands, and they are Ki67 negative, meaning they're not proliferating, meaning our definition of a dormant or quiescent cell. So in experimental animal models and in some human samples, we are getting a sense that there are some molecular pathways for tumor cells to find their way into the bone marrow and, in fact, be quiescent. So, of course, that's quite interesting, and understanding that is a starting point for doing something about it. And what's quite interesting about these niches is that because they're in the niches, they have some properties that are different than the typical tumor cells. Now, on this slide, you can see, as I point out, that they have a temporary growth arrest. Rather than proliferating and growing and being Ki67 positive, these cells tend to not grow and be Ki67 negative, but they tend to still be alive. So they're not dying, they're not going apoptotic like a leaf falling off the tree. They also have, like in the purple arrow, features that allow them to survive without actually dividing. And we can imagine many cells in our body like that. Many people will look at their brain or their heart of cells that reside for years, decades, but don't really divide very often. So there are definitely features of cells that allow this long-term survival. But more importantly, on the bottom side, there are some very good examples in leukemia that, and perhaps emerging examples in breast cancer, that these cells that are disseminated, that are long-term survival cells and don't have the proliferative capacity or the on switch for growth, they are resistant to chemotherapy and potentially resistant to hormonal therapy. And in our interest in immune therapies, they potentially might be resistant to being recognized by the immune system and having the immune system destroy them. So, as I said, if we are going to observe this phenomena and we're going to understand this phenomena, one of the things that we want to focus on is a process in which we can identify a pathway to eliminate them, and whether that's with conventional chemotherapy or hormonal therapy, that's one pathway, but we also want to consider the opportunity to have our immune system recognize and destroy these tumor cells. So what I'm showing here now is some of the pathways involved in immune recognition by the T cells of our body. T cells are generally considered the warriors, if you will, of the immune system. They recognize and kill abnormal cells, either tumor cells or virally infected cells. There are other warrior type cells that are helper cells that are also recognizing antigen. And this cartoon just shows you that there's a molecular process in which recognition occurs. And as you can imagine, if you had a molecular process that minimized the ability to be recognized, you might in fact have a immune surveillance strategy that allows you to go stealth, if you will. And there are tissues in the body that are slightly abnormal, that have a stealthy-like, or we call it immune-privileged location. And in fact, parts of the bone marrow, stem cells, if you will, are considered immune-privileged. The testicles and the production of sperm is something that is immune-privileged, because we don't want to have abnormal proteins produced by sperm being recognized as foreign and so forth. So the body does have processes that can affect these immune recognition pathways. We also know that in addition to immune recognition, there are co-stimulatory signals that are necessary to activate an immune response. So even if the tumor cells maintain their recognition machinery, activation pathways illustrated in this cartoon could be abrogated or modified, preventing the immune system to efficiently eliminate these disseminated or metastatic, micrometastatic lesions. And in fact, rather than just being speculation, there's very clear evidence now that immune modulation does occur in cancer cells, and this form of immune modulation occurs during the activation or priming phase of the immune system, shown on the left, as well as when T cells are ready to go and ready to kill tumor cells at the effector phase of the immune recognition and killing cycle. Immune modulation could occur. And the new therapeutics that are so exciting in the oncology field, these are called immune checkpoint inhibitors, and they eliminate the immune modulation due to CTLA-4 or PD-1 or PD-L1. These are having massive effects in many types of cancer, including lung cancer, bladder cancer, renal cell carcinoma, lethal forms of skin cancer, malignant hematologic malignancies. So a broader range of cancers appear to be using these immune modulatory pathways to avoid the immune system, and we suspect that perhaps breast cancer itself is doing this. So again, the idea that any one single therapy is going to be effective is something we can only dream about, but realistically we recognize that in different types of tissue, for example the breast, which in the life cycle of many women is required to make a protein that is passed on to a child for their nutrition, and this child will also receive nutrients as well as immunoglobulins. So there's a very complex immune regulatory environment that allows a child to get immune cells and immune proteins from their mother. We may not have a complete understanding of the immune regulatory mechanism associated with the breast, and perhaps this is one of the reasons that the immune modulators that are working so well in skin and lung cancer may need to be slightly different for breast cancer. So this slide just shows that there's a complex population of cells that all can have an impact on immune recognition, and we're looking and exploring the variety of cells and their various combinations to understand what will be required for the immune system. What are the potential therapeutics? As I mentioned, antibodies targeting these immune checkpoints. Again, the checkpoints work by blocking T cell function, so we may need to get more T cells around these micrometastatic lesions. And again, some of the T cells are warrior T cells, but some of the T cells actually are peacemakers, and getting the right type of T cell into the right location may be really important. And then finally, there are other cells in the body that can modulate the effector T cells, including macrophage and other forms of antigen-presenting cells. So the complexities and opportunities afforded by the immune system may have to be systematically and methodically applied. And this, at the molecular level, provides some evidence of some opportunities in which new therapeutics, heretofore not shown to be effective as single agents, but in fact may be critical in combination to eliminate some of these micrometastatic lesions. And again, a quite interesting opportunity in that very large, very big tumors that are causing symptoms and potential death, the treatments and the side effects of those types of treatments are clearly being studied, but the potential to eliminate these micrometastatic lesions and to eliminate them using these agents alone in combination may require a much more modest dose or a different timing, and so they're exciting opportunities to explore to prevent that. So let me finish up by describing our interest in the immune system and thinking that, of course, if we had, for example, a vaccine that would prevent these micrometastases from re-emerging or eliminating these dormant cells, we would provide perhaps lifelong resistance to these recurrences. And again, the idea that we can develop immune recognition strategies targeting these disseminated tumor cells is something that we're quite excited about. One of the problems with the immune system is that if you have a target, the tumor cells try to escape immune recognition by eliminating that target. So if you have, I always tell folks, if you have a rear view mirror that's green, and I say I'm going to eliminate all the cars that have green rear view mirrors, you might say, well, I'll just take the rear view mirror off my car and I'll be able to go on my way. But if I say, well, I'm going to really eliminate the fuel injector or the carburetor on your car, something that's essential for your car to go, that's not anything that you can remove. And again, the same thing applies to our strategies in understanding what are the immune targets that we want to go after. How do we eliminate something that even if the tumor cell wants to escape, it's not something so trivial like a rearview mirror that it can go on its happy way without really some dire consequences. So in general, immune strategies are emerging to be profoundly effective in metastatic disease and potentially in large locally advanced diseases. They do have some side effects, and the long-term consequences of immune strategies may not be as well appreciated as we would like, so we're cautiously optimistic about their use. They're being used in a variety of tumors, and we think it'll just take some time to understand how best and what are the right combinations to use in breast cancer. But we are also trying to dig a little bit deeper in considering, are these immune-based strategies that have the potential to create lifetime protection against disseminated or micrometastatic disease, are they an opportunity to intervene, and can the disseminated tumor cells really be recognized and be destroyed by the immune system? So again, for those who have an interest in some of the scientific things that we're doing, this is an example where we take a model of breast cancer in an immune-competent animal. The previous study I showed you that was on the cover of Science Translational Medicine, those studies were all done in mice that had a non-functional immune system. So we did those studies in the absence of immunity. What we're now doing is redoing many of those studies in animals that have an intact immune system and trying to understand how to manage immune recognition and destruction. And this is some unpublished data, just to give you an example of our ability to take bone marrow from mice that have metastatic lesions. In those pictures on the bottom, you can see the metastatic disseminated tumor cells, pointing to that little arrow in that middle column 2. And what we can do is identify these micrometastases in the bone marrow of mice. Again, quite a remarkable achievement. And again, in this next slide, we are demonstrating that these cells maintain the expression of a target antigen, and then we have very exquisite immune reagents to kill those tumor cells. And we're currently performing experiments with the support of the National Breast Cancer Coalition to identify how effective these cells can recognize and kill these disseminated tumor cells that are residing in the bone marrow. So we recognize and understand that metastatic cancer remains a clinical challenge, and we're entirely supportive of the ongoing efforts by many people to think about treating those metastases, and we hope that effective therapies continue to be developed. But we also understand and recognize that sometimes the most effective form of cancer therapy is in the setting of minimal residual disease and micrometastatic disease. And so our interests here are to try to develop very strategic plans to develop a relatively non-toxic lifetime protection against those recurrences, because we think that this ongoing persistent risk of recurrence and death for even the most benign, if you will, molecular subtype of breast cancer, the ER, PR positive breast cancers that have excellent 5-year survival rates, we think that the long-term ongoing recurrence rate is really unacceptable, and finding a strategy that's not just additional chemotherapy or lifetime hormonal therapy to prevent recurrences is something that is really needed, and we're quite excited about the opportunity to really make some headway in this area. So I wanted to finish up by recognizing that this is work that's not done by any one individual, and I wanted to really point out and thank those colleagues, Zach Hartman and Dorothy Simpkins at Duke, and then an individual, Cyrus Gajar, who is a terrific younger investigator at the Fred Hutchinson Cancer Center in Seattle, Washington. So thank you very much. I'm welcome any questions, and happy to answer or comment on any things that are brought up.
Thank you, Dr. Lyerly. I'm going to encourage people to ask questions by submitting them through the question pane on your computer screen. It should be on the right. In the meantime, we have a couple of questions. So do all long-term cancer survivors that are free of relapse also have dormant cancer cells hidden somewhere?
Well, that's a great question. I think we don't know that. We do know that amazingly enough, all people with initial diagnoses of breast cancer, if they are evaluated by doing a bone marrow biopsy and looking for disseminated tumor cells, a shockingly high number of people will have disseminated tumor cells, maybe 5% or so. And although you might then assume that, well, of course, all of these people are going to die of metastatic disease, amazingly enough, there is an increased risk of dying of metastatic disease, but not everybody with these disseminated tumor cells, in fact, dies or has a recurrence. And again, the hypothesis or question is that does everybody have these and they're held in check, or does it really exist where once you've been cured, you've eliminated every tumor cell from your body? And as the tools get better, if we look at the ability to look at micrometastases, circulating tumor cells, circulating tumor DNA, and so forth, there's a tendency to believe that there may be a state where long-term survivors have zero tumor cells, but there's another perspective, which is many people, even though they don't have any clinically evident disease, may have these dormant tumor cells in their body somewhere.
Thank you. So you mentioned that some believe that periods of stress can lead to recurrences, and the listeners want to know if you're talking about regular life stress, like financial concerns or a really long to-do list, or are you talking about a different kind of cellular stress, and do you believe in that yourself?
Sure, that's a great question. And so one of the things that we'll be learning is what are the activators of the progenitors or stem cells or potentially dormant cells. And some of the activators that we know of are periods of stress. For example, of a fracture of somebody's bone. Let's say they're going snowboarding and they break their leg and they had disseminated tumor cells. Do the signals and the repair mechanism and that level of stress lead to an environment that is going to promote tumor growth? As a matter of fact, it does. As a surgical oncologist, I can tell you that as we operate on women that have cancer, we're very sensitive to either making sure that we remove areas of scar that we've created during the biopsy period, or we make sure that scarred or healing tissue is incorporated into a radiation field so small deposits are not found. And oftentimes, if we do see a recurrence, a rapid recurrence, it can be in the setting of the surgical wound. So we know that there are some signals that lead to that. Now that's one extreme, and what we don't know if the extremes of stress that lead to your adrenal glands being stressed out or neuro connections between your brain and your bone marrow can tip the scales to trigger metastatic lesions. But we have, again, we have increasing evidence that the profound neurological stimuli can have effects on your body. There's not really in the cancer field, but there's a sense that profound sadness can cause cardiac dysfunction, where your heart literally begins to beat much, much less efficiency. You can go into heart failure from profound sadness. So we know that our understanding of what's going on in the body is imperfect, and we'll start out with some very profound stressors to see if they will have an effect on reactivation. And then obviously, as the tools get better, we'll look to see, is there a threshold effect in which types of stress are important to avoid for our health. I'd like to be able to say we should all avoid these things, and I try to encourage people to avoid those things, but that's always easier said than done.
So another question, how do you know that a new primary is not actually a metastatic recurrence? And does a second primary create a more favorable environment for dormant tumor cells to deactivate?
That's a fantastic question, and I think that's actually one of the questions that I've had, and we're in plans to study that. What we're planning on doing is using a DNA fingerprinting to prove that the dormant cell is in fact a molecular brother or sister of the primary tumor versus a new primary that would be sufficiently distinct to convince us that it was not part of the original tumor. So as you know, there is a thing called an occult primary for breast cancer, where there are rare instances in which you'll have somebody with metastatic disease and you cannot find their primary in their breast at all, and people think that these are very small tumors that metastasize very early. And so I always would question, well, if somebody is 20 years out, how do we know that this is not just a new metastases with an unknown primary? And I think, again, the molecular tools will be definitive proof for that. But again, from what we know now, it appears that the molecular fingerprints appear very similar. But I share that questioner's perspective that do we know for sure yet? And I would say we have the tools to answer that, but we're going to have to subject some of these tumors to that type of rigor.
Thank you. Is there any reason now to find out if you have dormant cells in your marrow? And together with that question is there another method other than bone marrow biopsy that might be less invasive?
Yeah, so what people are doing now is they're considering strategies to non-invasively determine your tumor burden. And so I think obviously bone marrow biopsy is uncomfortable and expensive and can lead to some real discomfort even after the procedure is done. And individuals have been proposing molecular techniques, I would say cell-free DNA analysis, to look for circulating occult tumor cells. I would say that there are ideas out there, but I don't think there is any proven clinical benefit to either monitoring your bone marrow or monitoring your cell-free DNA today. But I think people are aggressively trying to determine if there are strategies that are better. And what I would also forewarn people is that just because they identify metastatic tumor cells in the bone marrow or in a lymph node in a patient, it doesn't mean that 100% of those people are going to have a poor outcome. And that's why I would caution people that what we don't know right now is which ones that are identified as disseminated, which ones will go on to become macrometastases and become lethal. We know, again, that you probably have a greater likelihood of having a poor outcome, but that certainly is completely the opposite of saying you definitely are going to have a recurrence and need treatment and so forth. We just don't have enough knowledge about that. But there is a sense that a significant number of people will have these, and you tend to do worse, but there's no agreed upon strategy. And in fact, even today, we don't routinely get bone marrow biopsies from our patients that are treated. So it's not sufficiently informative that your clinician is bereft if they don't ask you to undergo that procedure.
Thank you. We have a couple more questions. Is the mechanism of cancer dormancy different for different types of cancer and for different genetic mutations?
Yeah, very great and insightful question. And I would say one example would be some of the cancers appear not to have a state of dormancy, and perhaps some of those are the most lethal forms of cancer, like brain tumors, glioblastomas, pancreatic cancer, advanced lung cancer. Those cancers don't seem to have a a period of time where patients can go on and live for a long time and not progress. Breast cancer, it seems to have this phenomenon. So there are some people who feel that prostate cancer, particularly those that might have occult bone metastases. So again, I would say it's a great question. A couple factors that may play into it. It could be those tumors that tend to go to the bone marrow may be more likely to be dormant because of the role of the bone marrow in supporting hematopoietic stem cells. So maybe it'll support dormancy in stem cells, and that same nutrient mix will support dormant tumor cells. Unknown, but some people hypothesize that. It could be the hormonally sensitive tumors, tumors in which hormones are felt to have a profound impact on their biology, like breast and prostate, may be more likely to be dormant, and again, some of the other tumors that are not that clear. And then the third one, which is again, I think a very profound question: are there molecular features? And again, I think the example where the triple-negative breast cancers don't appear to have a dormancy profile, but again, it's not totally clear. But there's again the data showing that if you're alive at 5 or 10 years with triple-negative breast cancer, you're very unlikely to have a recurrence. But if you're alive at 5 or 10 years with an ER/PR positive breast cancer, you still have this unremitting, small but unremitting risk of having a recurrence and dying of breast cancer.
Okay, the last question. There's some research coming out of the University of California in San Diego, the Moores Cancer Center, that's saying that vitamin D and calcium in adequate levels is playing a role in potentially allowing a developed cancer to enter and stay in a weak or fully dormant state. Can you comment on this?
Right. Well, again, that's an outstanding group at the University of California in San Diego, and I think there are many different models in which a variety of manipulations are being attempted. So I don't have a detailed knowledge of that, but I would look at that and see what their model is and what the impact of those interventions, vitamins and calcium. Again, if you imagine that it's the bone microenvironment that is having an effect, and if a vitamin D and calcium are modulating bone osteoblast activity, and the stressors associated with bone remodeling are leading to signals, I mean, I could certainly see how it would make a lot of sense. And as I mentioned before, even breaking a bone and having that inflammation and signals of stem cell activation can have a profound effect. So those vitamin D and calcium effects are directly on the tumor cells, or perhaps in the environment of the bone marrow, I could certainly see them being important and playing a significant role.
All right, Ivis has one more question for you. Can osteoporosis treatment affect dormancy?
Great question. Again, we don't know, but it may be. With these models, we're hoping to explore them and see where they're going. So I wanted to thank everybody for participating in this. I think these questions were terrific, and I think it's exciting to think that there's a lot of interest and enthusiasm in this area of research. I want to thank all of you for participating and really for the support and guidance that you provide us as investigators and clinicians in really tackling this question that's so important to all of us. Thanks very much.
Thank you for an incredibly informative program, and thanks to everyone for participating.